So I just want to reiterate what I wanted the main message to be from all of that. That we need to collect a lot of information about the vaccine, about how it was given, about the person who got it, their past medical history, things like that. We also need to collect information about the other people that got the vaccine and other people who didn't get the vaccine. All of this type of information, which we collect to fill out in these forms, which is being done, is all taken into account when we try to figure out if the vaccine caused the adverse event. If you don't walk away with anything else from today, walk away with that. But up to now, we've only, all of that has actually, all that work was really, that we've talked about, was to decide whether or not this occurred in that individual. So I want to spend a few minutes talking about what we do, how we think about causality when it's been reported in more than one person. And I'm going to try to touch on it just quickly, and maybe you can think about this as a preview to next week, because this is really, again, at the heart of what's next week when you talk about causality. So if you don't absorb a lot of it, at least when you hear it again, I'm that kind of person that when I hear something a second time, I learn it so much better. So if nothing else, just say, okay, now I've heard it, then I can really get it next week. And then I want to go through an example which may help. So we left it that we had to do all that work for that one case. Now let's move on to the situation where you have a second report come in with the same adverse event. So now you start to have to think a little bit differently. You think about, could this be a signal? That's the word we use. Signal detection. This is when signal detection really comes into play when we're talking about more than one person. I just have this slide because as I mentioned, this special consideration we have in the world of vaccines, that we have immunization programs and pharmacovigilance centers doing similar and sometimes collaborative work. Sometimes we use different words. When we talk about AEFIs in the WHO document, they use the term cluster, which is a cluster is 2 or more cases of the same adverse event related in time, place, or vaccine. So more than one case. We don't use the word cluster so often when we talk about it in more routine pharmacovigilance. We tend to talk about signals. This doesn't have to do with how many cases you have, although we think about it, it has to be usually more than one case, but it's reported information on a possible causal relationship between an adverse event and a drug. Usually more than one report is required to generate a signal. So there you see. This isn't in the official, this is a disclaimer to this. You don't always see this, but I thought it was helpful here to think about clusters and signals. I think we're referring to the same thing, but one of them might be an immunization program term and one of them might be a regulatory term. But I want to challenge you to think that they're very similar. They're very similar, and the way we deal with them, we actually use different language as well. Immunization programs usually use this cluster analysis. In more routine pharmacovigilance, we talk about case series causality assessment. But they're very similar processes. So I think that in AEFI analysis, a lot of the cluster analysis algorithm that you may see is really focused on trying to figure out if this is something related to the vaccine process, the injection, the vaccine itself, was it kept at a cold chain? A lot of known reactions, trying to figure out if these expected adverse events, both those that are mild, but maybe these that are more serious, like BCG, disseminated BCG, those types of things that are known, we're trying to figure out if they're occurring more often than normal. Case series causality assessment, on the other hand, which we talk about more routinely, I would say that this is more what pharmacovigilance centers do when they're trying to figure out, is this a new signal that maybe we don't understand yet, that we can only detect when we give it to a whole lot of people? Maybe we think about it in this way when we're trying to define new problems. These to try to quantify if something that's known is occurring more often. I'll leave it there. This immunization program's identifying clutters. There's another algorithm. And as you see, we go through some steps, and it gives you a lot of endpoints where you're trying to figure out, was this related to getting the vaccine? Is it a batch problem? Is this a known reaction? So I think this is very helpful for trying to go through when you have more than one case to go through this algorithm. I'm not going to go through the details because I really appreciate now that not everyone is doing this work routinely. But just to know that when you have more than one adverse event and you need to quantify, and you're trying to figure out if there's something, the same issue is causing multiple people to have a similar adverse event, this is very helpful. Batch-related problems, injecting wrong. Now I'm going to move over to maybe identifying new signals. This is the part of the talk that you'll hear again next week. So this is the more pharmacovigilance, more routine pharmacovigilance language that we talk about trying to identify if I have a signal, if I have a number of cases that are reporting the same adverse event, how do I assess causality? So not at the individual level, but I have a number of cases. So, causality assessment, this is a drug article that reminds us that the way we do causality assessment of a collection of case reports or of a case series is that we apply the Bradford-Hill criteria. You're going to hear about him again next week. But I thought I would just mention him. You can't have a talk really about Bradford-Hill. I had the opportunity to go to the UK to get a lot of this education, and the people from the UK, they like to put up pictures of, these sirs that lived before, and they elucidated this thing. So I do it. So it's very not American, but it's very British, so I do it. So he's the man who is essentially credited for finding the link, for establishing, for just elucidating the tobacco and cigarette smoking can lead to lung cancer. And when he did that, he established these criteria that are used for causality assessment, again, with a group, not at the individual level. So, and he has these criteria. Again, I don't expect you to get it now, but you're going to hear them again next week. But just so that you hear them for the names, and I put them up here because I wanted to just point out that, you can think about this in terms of vaccines too, but the strength of association, the numbers of cases you have. If you only have a few reports, it's less likely to be a causal relationship than if you have a lot. So when you start to get larger and larger numbers, you get more suspicious. Consistency. One of the things that we say in pharmacovigilance, if you have the same event reported from distinct locations, it's more likely to be something going on. For vaccines, from a global perspective, again, looking at rare adverse events, things that may not be known. The pandemics and narcolepsy example I give you because it's the one that I essentially lived through, was that this was a signal that was seen in Finland, totally unexpected. Some children started getting narcolepsy. We saw it in Sweden too. That was very strengthening the potential for causality. It wasn't just one particular population and one doctor making a diagnosis of narcolepsy. Another doctor in another whole country was seeing the same thing. So it can be useful for vaccines. And again, I'm not sure that would have necessarily been picked up at a more local level going through AEFI cluster analysis. It requires a global, more general pharmacovigilance perspective. Specificity, some adverse events cause ADRs by different mechanisms. We'll talk. I didn't give a good vaccine example here, but acute renal failure can have many different flavors, I like to say, pre-renal, post-renal. So if you see the same type of renal failure, pre-renal, it's always pre-renal failure when they report acute renal failure with that drug. It's not sometimes interstitial nephritis. So a specificity if it's all very similar. Temporality, this is the one, that most of our suspicions about adverse drug events and their relationship to drugs we have, because, usually patients only report an adverse event after they took the drug. But also a reasonable time to onset. We tend to give more credit if you have a population of people who report the same adverse event within, the same time span. If you have one that reported after 1 day and one person that reported after 1 year, it's a little less likely to be causal. Biological gradient, when we talk about normal drugs, we talk about with increasing dose. With vaccines, there's a slight different version of that. Usually we give vaccines in multiple doses. It requires several injections to get the full immune response. So we look to see if we have similar adverse events after subsequent doses. That can be a sign that it could be something. And then theoretical possibility, this we saw in the individual case causality assessment. That's the thing that I think when we talked about much more information is required at the individual level for vaccines, we really only start to talk about that when we have more in more routine pharmacovigilance. But it's the same concept. In a way, you're applying a bit stricter criteria that you're already looking at that at the individual level for vaccines. But again, it's the same types of concepts. Coherence, that you don't propose an adverse event is caused by the drug with a pathophysiology that's completely the opposite of how the drug really works. Experimental evidence, animal studies, and those types of things. And analogy, so you see something similar in another product that's similar. So small fiber neuropathy, it seems like a very specific thing, but at one point I was looking at that, suspecting that I actually had a case report of that after a vaccine, and I looked and I found a paper that had actually been reported with other vaccines. They were actually of a different nature, but they had done biopsies, and so I thought that lends a little credibility to the fact that it could have happened with this vaccine. So again, those are the criteria that you think about when you're trying to determine, when you have a collection of cases, whether or not there is a causal relationship. And the short and simple way of talking about all of that is to just say you're looking for patterns in multiple case reports. I mean, that's essentially what you're doing. How similar are these to each other? Are they reporting the same event? The same flavor of the event? Do they have a consistent time to onset? Did they get treated the same way? Sometimes when you don't have an exact diagnosis of something, if you see that they all got steroids, say they had some sort of interstitial pneumonitis, you don't really know if is it infectious, is it inflammatory? That might be the only term you get, but if they all got steroids and got better, well, that suggests it could be the same thing, even though it's not very specific. Keeping those types of things. So, those are the types of things to think when you have a stack of the same event. And now I have to say one other thing, which is a little bit more specific to vaccines, in the fact that it's talked about separately. We don't typically talk about observed versus expected analyses when we talk about signal detection for drugs. But this is a pretty big component of vaccine work, and you hear this language a lot. And I've never been involved in actually calculating this myself. This is routinely in the European system, this is routinely the job of the marketing authorization holders. We ask them to do this. But I have a feeling that you might have to be doing that type of work even when you're collecting these in the National Immunization Program centers. And a lot of it depends on whether if it's a known reaction or an unknown reaction. So essentially what you're trying to figure out is this event that we observed in this group of people, is that how many we would expect if nobody had gotten the vaccine? So we observed X number of cases of, it's so easy to use narcolepsy, I'm sorry. We observed, the way the story went here was, in Sweden we typically diagnosed one physician, I should say just for one physician. He typically saw one case of narcolepsy per year in children. And then in a 6-month period, he saw 4. So he expected 1, but he observed 4. So that was what triggered him to be worried that something was going on here. So When we talk about AEFI clusters, you see this concept of observed versus expected here in this part of the algorithm is the rate of reaction within the expected rate. And then just as I give you this, just so you're familiar to know it's out there. So, you know, they give you an example. This is nice reference at the WHO. It's a little dated. I don't know if they have a new one. It's from 2000. It's a little dated. But it gives what we know about common reactions after these vaccines, these known reactions, and how often they occur. Because we know these estimates based on clinical trials and from more studies that we have. So this is very important to say, because we expect a certain amount of fever. But maybe we're seeing more fever than expected. If you can remember the examples I gave with influenza vaccine, there was one vaccine that gave a lot more fever than was expected compared to all the other ones, and they found that there was something related to the manufacturing. The important thing to know is that you can find information on what that background rate should be, because you have to have something to compare to. You're collecting the information on observed, and you need a reference for what you would expect in the background. This, again, for things that are known. We do it for things that are unknown, and this is where it's very hard. To be honest, there wasn't a lot of studies about how common narcolepsy was. This was anecdotal. This doctor said, well, I normally see 1, and I got 4. But I think one of the things that we first had to do, we had to establish how often is narcolepsy diagnosed. And that happens a lot. Right now, there's an ongoing signal for HPV vaccine in Denmark, at least. It's still ongoing there. But there's been a diagnosis made of postural orthostatic tachycardia syndrome. And there's not a lot of good data. We don't really know how often that occurs. Very scant literature. So we don't even know what the background is. So it's very challenging. A lot of countries, what they do when they're going to introduce a new vaccine on a mass scale is to try to make these estimations beforehand. They did this with the influenza vaccines during the pandemic. They went and they said, what kind of adverse events do we see after vaccines sometimes? We see optic neuritis, we see encephalitis, we see spontaneous abortions, we see Guillain-Barré syndrome. So what they do is they actually collect the data and say it occurs this often. This is what we have. And then when they introduce the vaccine into the program, they can see if what they're observing is higher than what they would expect based on what they've calculated before. And actually, when pandemics was being used here in Sweden, one of the jobs that I had was to kind of at least not on a fully statistical way, but kind of a little bit more. I was counting cases of Guillain-Barré syndrome after pandemics because we knew that a vaccine against swine flu previously in the United States in the 1960s, 1970s had a real increased rate of Guillain-Barré. So that was one of the things that we were kind of worried about. So I was counting the cases that we were getting in Sweden as they came in, comparing it to what we knew about how often it occurred. So I knew when to start getting nervous. That ended up not being anything, but we got pretty close. And then we did a subsequent study and found that there was maybe only a mildly increased risk, but it was nothing of the magnitude that it had been in the United States before. So, follow up. So let's say we've identified a signal. We have a collection of cases. We think the causality is suggestive that there is a relationship. And when we do observe versus expected analyses, we're a little suspicious that we're seeing more than we should have. What do we do? This is the real hard part. And this is the hard part because things with vaccines are so sensitive. Because we want to ensure confidence in immunization programs. So we're really careful about what decisions we make. Do we communicate this signal? Do we do regulatory action? That means like change the product label. Do we include it as a possible adverse event? Do we write healthcare professional letters and say, we've observed this. We don't know if there's a causal relationship, but be on the lookout. Or do we go so far as to say, we need a study. This is a big enough concern. we should arrange for an epidemiological study to actually estimate, to see if there's an increased risk, and then to estimate how big that risk is. This is just some examples in the United States. They actually have a program where they already in place to respond to things like this. Vaccine Safety Data Link, I think, is the old name. They have a new name called PRISM, but they're primed with their computer, everything, their insurance programs and diagnosis code to be able to respond, to be able to do some sort of observational study based on existing data structures to investigate some of these signals subsequently, to be able to make these estimations of risk. That's quite advanced, I would say, of course. And then I just put this on here, our little lonely signal document. Because, even if we don't do a study, or even if it doesn't end up in the label, it may be worthwhile to communicate. We have seen this. We don't know, but be on the lookout. And I mean, a signal still is just information about an association. It doesn't imply causality. But sometimes it can be helpful to communicate a signal, even if causality isn't firmly concluded. So, I have 30 minutes for the example. And I think we can, I had meant for this to be a little interactive, but we can see how it goes. So, this is a true, I've changed this case a little bit, but it is a true situation. And if you get the signal document and you've seen it in the last year, you may have seen something about this. So you kind of see what the endpoint was. So, say you get your first case comes across your desk. You work in Denmark, let's say. A 3-month-old infant from Denmark received their first dose of pentavalent pneumococcal vaccine. 21 days after the receipt of vaccines, the patient experienced a blistering rash, which began on the palms and soles and spread to the trunk. The patient was hospitalized. A skin biopsy was consistent with bullous pemphigoid. The patient was treated with steroids and improved. The case was classified as serious because the patient was in the hospital. There was no reported errors in administration or in the vaccine. There was nothing obvious. There was nothing obviously concerning at the time that the child got the vaccine, with the vaccine or the process itself. So my question to you is, should this case be further investigated? Yes, it's serious. So that was kind of why I put that statement in there. Okay. And then if yes, what type of information should be collected? This is getting back to that AEFI investigation form. So this very arduous, laborious process of gathering as much information as possible about the vaccinee, past history, do they take any other medicines? Have they experienced anything like this before, without getting, before they got the vaccine? Also trying to get information on the vaccine itself. It's been reported a little information. Yeah, the bottle looked fine. There was nothing unusual about administering it. But also, did any other children in that same setting, did they get bullous pemphigoid? How often does bullous pemphigoid occur in 3-month-old children in my setting, or is there any literature from anywhere? That kind of information. Again, pretty detailed at the individual level for a vaccine, and maybe some of these things about how often bullous pemphigoid for another medicine, a more commonly taken, more traditional thought medicine. Maybe you wouldn't collect that information till later. You wouldn't start thinking about how often does that occur. But start thinking about it now for vaccines. Do you have any initial thoughts on how this adverse event could have been caused by the vaccine? If it was. We haven't established that yet, but just to start thinking about it, hypothesizing about it. That's where a little bit actually doing some of this investigation early on helps. Learn about bullous pemphigoid. Who does it normally happen in? What's its pathophysiology? So this is rather rare in children. It's much more commonly in elderly people. That's the thing to start triggering me to think, that's weird. That's not what I expect. I don't expect it in a 3-month-old, in a normal. So it's enough to kind of think about it. It's nothing that necessarily is commonly reported after vaccines. Local reactions are at the site of injection, but this is different. This started on the palms and soles, at a site distant from where they got vaccinated. So it's a much more systemic thing. So when I think of that, I think a little less likely about injection-related, and I might think, is this child predisposed to something? It's a little more vague. And then if you go a little further, you find out that it can be it can be a manifestation of autoimmune process. So then you're thinking on a different level than related to the injection itself. So here's the adverse event form. This is just to remind what we talked about. You're collecting information about the patient, all the documents. This is good. I reported to you the biopsy results. That is very important here. That would be one of the tests and that kind of stuff, that extra information samples. I don't know if you need the biopsy itself, but a biopsy report. It's good to establish the diagnosis. This is truly bullous pemphigoid. So, kind of going through that AEFI thing, we got to that part again, if you remember, we were trying to decide if this is a true diagnosis. We're looking at the eligibility. And we said you have to identify the person, the vaccine, the event, and then we're trying to harmonize the definitions. Remember, we talked about the Brighton collaboration. The Brighton collaboration doesn't have a definition for bullous pemphigoid, but you can find something. You know, I just looked to see diagnostic criteria bullous pemphigoid, and you can do this and kind of get an idea. So there we go. We just go through this. I don't want to go through all these details again, but go through these questions again about causality assessment. So this kind of leads me to find out more about the disease. When I ask, is there evidence in the literature that this vaccine can cause the reported adverse event? Well, I can tell you there's not been any studies done. There's not been any studies done to find out if a vaccine causes, no epidemiological studies. This event is so rare. It's 1 out of 10 to 1 out of 100,000 people. There's no studies that prove or disprove that, or at least try to estimate the risk. So we find out this initial information, and as I mentioned before, it primarily affects the elderly. And as you can see, I mean, there's kind of a range. This is kind of getting an idea. Again, part of this initial thing that you hinted at, it's a lot of information, but how commonly does this occur? Well, I found in the literature that it most commonly occurs in the elderly. And there's a kind of a range there in different countries in Europe about how often it occurs. But it occurs on the magnitude of a few cases out of 100,000 to 1 million. That's pretty rare. What typically trigger bullous pemphigoid? Malignancy, recurrent trauma, systemic disease, psoriasis. And I'm thinking back to all that information I collected on my infant. Did that infant have any of this history? Well, I don't know if they had recurrent trauma, but they had some form of trauma. They got a vaccine. That's a mild form of trauma. And then if you actually find a paper of bullous pemphigoid in infancy. And this group in Israel had found one case, and they went to the literature and they found 79 other cases. So they decided to describe them all. So this looks very characteristic, blisters on palms, soles, and face. That's what happens in kids. That doesn't actually happen in the elderly so often. And then they actually estimate an incidence of 2 per 100,000. in children less than 1. So there's kind of an idea of what your background rate might be. I think this is I don't have I do not have an answer on whether or not a vaccine causes. I left it general for vaccine because I'm not sure that it's related to the specific component, as well as maybe more the risk is just getting an injection, whatever it was. But I just think this is very interesting. So I can't tell you that I think that the vaccines caused bullous pemphigoid. But I think this picture is incredibly interesting and leads me to believe that there's a possibility. These spikes, 2, 3, 4 months. It's just pretty striking when I look at it, if it's possible, that's almost coincides a bit when children get immunizations. It could be any injection. I'm not saying it is related to the vaccine, but they do get poked 2, 3, 4 months. And again, just a little bit about pathophysiology. This is important when you're trying to answer that question on that investigation form, is it biologically plausible? You need to kind of understand how the disease was caused. So, and it is an autoimmune response, and it appears that inflammation in the skin could lead to a disruption of the basement membrane, and you have immune complex deposition. So, probably not caused by the vaccine per se, but could it have been that any injection, any trauma to the skin could trigger this in someone who's genetically predisposed? I'll just throw that out there. So, has this been reported before? As I mentioned, there's been no epidemiological studies for something this rare. So I don't find any meta-analyses or epidemiological studies, but I find a lot of people reporting this. I actually had this case about a year ago, and I'd never heard of it before. And I find all these journal articles, bullous pemphigoid in infancy developing after post-vaccination. And I see this term, post-vaccination bullous pemphigoid in infancy. And then another again, post-vaccinal. And they're all in dermatology journals. And I'm thinking, that's just amazing. Again, I see a lot of case reports from all over, not just limited to one country. So that's a bit interesting to me. Again, kind of thinking back on the Bradford Hill, just to try to start you thinking about it. You know, I mentioned before, you're a little bit more inclined to think that something is possibly causally related when it's reported from more than one site. So if I see a bunch of case reports from different parts of the globe. But I think what really struck me when I reviewed this, when I was looking for these reports, was that the dermatologists already had a name for it. But it's not included in a label. No one's done a study on it, but they acted like it was pretty routine. So I'll skip that since we're a little short on time, but the question's related to that. So how would you assess the first case, individual? So we've done all this research, we've collected all this information. Just kind of think. I don't want to, again, we don't do this all the time. In this case, is there strong evidence for other causes? Well, we know we have a diagnosis of bullous pemphigoid. We don't really know how it happens, though. I'm probably a little reluctant to go straight to inconsistent. But I'm also a little bit, is there a known causal association with the vaccine? No, not really. Not that we know that's been proven. But we have some case reports. So these are always good, but you don't always, kind of just as you mentioned before, sometimes it's hard to go through this. Either because you don't have the information or because no one has the information. We just don't know. Is there strong evidence against a causal association? Well, there's certainly not a study like the MMR and autism that says this is not related. So review qualifying factors. This is where you're in that realm where you're kind of thinking about the Bradford Hill criteria in my mind, you know. So, I mean, for me, this is a bit indeterminate. If you look at indeterminate means that you can't rule it in, you can't rule it out. These are potential signals land here. So then what do we do? This is the hard part. Well, I'll tell you what I did. I wanted to see if there was other cases, because I was pretty suspicious there was. So it's been reported in the literature so often. So in this, again, is where it helps to have a global database, perhaps. Probably most regional, individual regional centers, they may never see a case of this. I might see a bunch, because I'm looking at the vaccines for, the whole program. So based on what you learned yesterday or the day before from those people that would have access, where would you look? You have any ideas? I went to look in our Vigibase in Vigilize. So I hope you can see this. I didn't know if I'd have internet access, but so here's the Vigilize. So this was initially reported. It was reported with pneumococcal vaccine and pentavalent vaccine. Just to make it simple, I decided to choose pneumococcal vaccine. I will say all of the cases, they got both of them, but I just tried to make it easy. This is kind of routine. I would call this routine immunization, because I'm not really suspecting that it's this particular vaccine, so we're saying. So I had 12 cases with all that actually reported bullous pemphigoid. So they had actually had most of these were pretty sure diagnoses. These weren't just rash. This could be, you could expand this. There's potentially a lot more cases that didn't get reported as bullous pemphigoid. They could have just been reported as a blistering rash. So this is a very narrow view I did to make easy. But you'll think about that next week. We'll go through that exercise. Just because someone didn't get, there may be many people out there that just didn't get that diagnosis, but they had a blistering rash on their palms and soles. I didn't even catch that here. Here they are. I thought this was nice. I hope you can read, but I did this just so you could see mostly. This is all the cases. As you can see, they were all serious. Most of them were in the hospital. These are the types of things, again, to think about are the patterns. Yeah, the pattern is it's usually serious. They end up in the hospital. The geographic distribution, I have Sweden, Japan, Denmark, France, Ireland, Czech Republic, United Kingdom. When you look at all of these cases, you see that there's their ages. They were all classified as serious. They originated from 8 different countries. Time to onset ranged from 5 to 21 days. One report noticed a positive re-challenge. You'll hear a lot more about that. But so this essentially means that the child got the vaccine, got the rash, it resolved. They got the second dose, it happened again. These are the kinds you really hang on when you find. Now, 2 reports didn't have that. So that could be just as important. 2 of them didn't do that. But this at least implies that it's plausible in some people. So it makes it very hard for me to rule it out completely. Again, this was just to say that this is very similar. This is a similar type of thing. I just put this here. I only had single case causality assessment reported to us for 5 cases of the 12. And it's a bit interesting. Almost certain it was written, probable, and 2 possible. So you see, there's a variety. Different people assess things. Could have varied on the amount of information they had. So when you're up at the case series level, you may have a total spectrum of how people assess causality at the individual level. But I thought it was pretty interesting that one said almost certain. So looking for patterns, that's kind of what I was trying to do. Where did they come from? Did they all have the same? Did they get treated the same? Were they all serious? Were this a rash in the same place? Oh. So what would you like to do? 12 reports. And again, the decision that was made at the global level may be very different, because this is probably not a situation many regional centers are going to be in. They're not going to get they're not going to see 12 cases of bullous pemphigoid. Should I keep the cases and re-review? Should I change the product label? Should I get somebody to do an epidemiological study? That would probably be not very feasible to do that, because to detect something that only occurs in 1 out of 100,000 or so would be very hard to do. And we have only 12 reports. Now again, I said there could be many more, but I have a very small number of reports, and I know that pneumococcal vaccine is given to all kinds of people. So this isn't occurring very commonly. But it could be that in the few that are disposed, it could happen. So the decision, I thought, the kind of middle of the road, I thought, was to communicate this as a signal, not to say that the causality is established, but I thought an important public health intervention would be to inform people that are involved in vaccines about this phenomenon that's been observed, and that it seemed like it was very limited to the dermatology realm. And not everyone can go to the dermatologist, and that's probably not the point of entry for a lot of these patients. Not to say that causality is established, but to make people that are seeing these patients in pediatricians' offices and vaccination centers aware that this has been reported. We have a number of cases. And the reason being because these children actually require going to the hospital, and they require steroids. And also because they're going to need another vaccine. Most of these vaccines you need to take repeatedly. So these types of things, just to make people aware. Again, we're not sure, but if you come across a case, you might need to think a little bit about it. So, because again, a signal doesn't establish causality. It's not saying causality is established. It's just something to be aware of, that maybe we can minimize risk if we know about it. So take-home messages. Vaccines are one of the greatest advances in public health and have brought enormous benefit to many. I should say this is also my little personal mantra about vaccines, but I hope I've tried to hit all these. However, vaccines can cause harm at the individual level by a number of different mechanisms. There's an increasing shift in the burden of vaccine pharmacovigilance to lower and middle income countries as new vaccines are being directly introduced into these countries. Close collaboration between national immunization programs and regulatory authorities are required for successful vaccine pharmacovigilance. Increased cooperation and pooling of data better allows for the detection of rare adverse events, which can be significant given the extensive use of vaccines and the delicate nature of vaccine safety concerns. And in the future, pathogenesis of adverse events following immunization will likely become better understood and linked to specific genetic profiles. And current paradigms of vaccine-based population may need to adapt accordingly to incorporate individual exclusions based on genetic profiles as progress in vaccine safety is made. So that's what I hope I have been successful. I know that this has been a bit complicated, both for a variety of reasons, but I hope you got something out of it, and please feel free to come and ask me any questions. And we finished a little early.